Hi, welcome back. I'm Wei from Scientific Computing Department at TFC. I've been chairing this section for the first 3 talks, and then I will hand over to Stefano for the last 3 talks. The first talk will be given by Sofia. She's going to talk about how can emerging computational approaches help us understand drug resistance and allostery in proteins. Welcome.
All right, let's start then. Let me just try this. Brilliant. So I'm Sofia. Well, hi everyone. So I'm Sofia, and I'm a BBSRC Discovery Fellow in the School of Chemistry at the University of Bristol. And today I'm going to kind of show you how an emerging computational approach can provide unique insights into drug resistance and allostery in proteins. All right. Well, I hope, or I think that we can all agree here that proteins and other biomolecules, they're not static entities, right? Proteins are always moving and they're always changing conformations, they're changing their structure. But they also have a very unusual ability. Proteins have the ability to respond to changes in their environment by changing the structure, their conformation. And this is definitely a fascinating feature of proteins, but it's actually a bit more than that. This is essential for their function and is the basis for most biological processes. As such, I think it's fairly easy to understand why, in order for us to fully understand how a protein and other complex biomolecules work, we need to have a detailed description of their motions and understand their functional dynamics. Right. Probably the most common way to study, the most common approach to study the motions in proteins is through molecular dynamic simulations. And in this approach, we simulate how the conformation of a molecule changes over time. And in order to run a simulation, we need to first have the structure of a molecule of interest. So this structure can be determined experimentally using X-ray, NMR, cryo-EM, but it can also be a model produced by AI, by AlphaFold, for example. Besides this, besides the structure, we also need to have a mathematical function, an equation that's able to describe all the interactions or the interactions between all the atoms in our systems. You have here an example of a very common force field. It contains many terms. I'm not going to go through all of them. And then, by using classical mechanics and integrating the Newton's equation of motion over time, we obtain a group of conformations that a molecule of interest can adopt. But it's a bit more than that. We know, or we can know, how these conformations here interchange with time. And this provides us a trajectory in which we have a description of the motions in our system. Right. Let me now give you a very concrete example of a system in which its motions, they're essential for the function. Let me introduce to you nicotinic receptors. So nicotinic receptors are ion channels that are widely distributed in the nervous system, where they have many roles, and they're associated with several diseases and conditions, including nicotine and alcohol addiction. And in these receptors, the binding of small molecules, here shown in red, the binding of small molecules of ligands such as, for example, nicotine, the binding of nicotine to the receptors, for example, opens an ion channel down here. However, and this is where the problem lies, in these receptors, the ligand binding site, the nicotine binding site here is located here in red, and it's located in the top of the receptor. However, the ion channel is located down here and is highlighted in yellow. And this means that somehow the structural changes that are induced by the ligands, they need to propagate throughout the protein structure to reach the ion channel. Right, as I said, the most common tool to study the motions of proteins, of biomolecular systems, is equilibrium molecular dynamic simulations. And equilibrium molecular dynamic simulations by themselves, they cannot answer the question of how structural changes propagate. And there are reasons for this, reasons that some of you here are very well aware of. One of the reasons for this is that there's no driving force for the structural changes to occur in the systems because they are in equilibrium. And second is that for most proteins, for most complex biomolecules, we cannot access either the timescales or the number of events needed to obtain statistically significant structural changes. And this here are the reasons why there is still a need to develop new approaches, new computational tools that allow us to study how structural changes propagate. And given this, at Bristol, we've been developing an approach that by combining simulations in different conditions, in equilibrium and non-equilibrium conditions, we're able to overcome or at least mitigate the limitations stated here. And this approach allows us to map the evolution of the structural and dynamic changes occurring in a system. So this approach is called dynamical non-equilibrium molecular dynamic simulations. Well, it's a really long name, it's a mouthful, we know, but we actually call them DNMD simulations. And at this point, the DNMD approach is completely general, and you can run this type of simulations in any MD engine, so the one that you prefer the most. So we've so far used GROMACS and EMBER to run some of the simulations. We also have used OpenMM, and we're currently working on SHARM and NAMD. And what we do in this type of simulations, which are the first of their kind, is that we introduce a perturbation in the protein through, for example, the binding or unbinding of a ligand, or through a chemical reaction, or the introduction of a mutation. And then we see how that changes, how that affects the structure and the dynamics of the protein. So far, we've used the DNMD approach to study several different, well, fundamentally different biomolecular systems. You have here some examples. But I think it's really important to state that up until now, in every single case, we were able to obtain new and unique insights into the working mechanisms of these systems. And what I'm going to do next, I'm going to give you a couple of examples in which the DNMD approach was instrumental into obtaining these new insights. All right, so my first example today focuses on nicotinic receptors. And in this case, we use this approach, the DNMD approach, to understand how the structural changes that are induced by the ligands in the top of the receptor here, how these changes are propagated to reach the ion channel down here. And in this case, our simulations not only provided us with the regions in the protein that respond to ligand binding and unbinding, but they also provided us with the order of the events that need to occur for the structural changes to be properly transmitted to the ion channel. So in this figure here, we have the response, the structural response of a human nicotinic receptor to the removal of nicotine. And we map this response in the structure of the receptor using this color scale here. And in this color scale, dark blue colors correspond to no response, no change in the receptor, whereas red color regions correspond to the largest changes. Now, if you look at this figure, you can see that the first region to respond, to respond to the removal of nicotine from its binding site in this receptor is this region here, called the loop C. This is not surprising because the loop C is a small region of the receptor that covers the binding site. So it's kind of the lid that covers the binding site. So it's not surprising that it's the first region to respond. However, from this figure, we can also see that the structural changes that start here, over time, they're transmitted to other parts of the protein, such as, for example, this region here, which is called the loop F, and then to the M2, M3 linker down here. if we now visualize this exact same figure, but in an animated sequential way, you can clearly see that the structural changes in response to the removal of nicotine, the changes that start here, over time, they're transmitted downwards to the loop F and then to the M2-M3 linker. The M2-M3 linker is a small part of the protein that binds directly to the ion channel. Right, we also use the DNMD approach to understand how different human nicotinic receptor subtypes respond to the removal of nicotine. And the results were really interesting because we were able to see that despite the fact that the different subtypes, they do have different parent rates of propagation for the structural changes, the regions in the protein and the order of the events needed to transmit the changes to the ion channel, they're always the same. And this allows us to identify a general mechanism for interdomain communication in this family of receptors. Now, let me move to my second example, and my second example of the day is the SARS-CoV-2 spike protein. I think we've all heard about the spike protein, right? It doesn't require a big introduction, but in this case, we use the DNMD approach to understand how these fatty acid binding sites, here shown in red, that were identified during the beginning of the pandemic, that were identified in the top region of the ectodomain, of the head of the spike, how these fatty acid sites are able to control the behavior, the dynamics of functionally important parts of the protein. And in this case, our simulations indeed showed us that the fatty acid binding sites, here shown in the top of the protein with the gray, here highlighted with the gray spheres, this fatty acid sites, they can control, they can modulate the responses of important regions of the spike. Regions such as the receptor binding motif shown here. So this motif is the part of the spike that actually binds directly to the human ACE2 receptors. But we also were able to see that these fatty acid sites, they can control the responses of this region here, which is called the fusion peptide surround region. And this region, the fusion peptide, well, the name is a bit self-explanatory. The fusion peptide is a small part of the spike that actually fuses the host and the viral membranes. So this is a really important, functionally important part of the spike. We also use the DNMD approach to understand how mutations such as the D614G that was present in several of the variants of concern, how these mutations affect the internal transmission of structural changes within the protein. And for the D614G mutant here, shown on the right, we were able to show that the presence of the mutation severely decreases the transmission of the structural changes to the fusion peptide region. And we can see that very clearly if we compare the response in this region here, the response of the mutant protein, with the response of the same region here in the original protein, in the wild type, the protein without the mutation. We've also used the DNMD approach to understand how changes in pH are able to modulate the dynamics of the delta spike. And our simulations in this case revealed that a decrease in pH that is similar to the one that's observed inside aerosol particles, for example, this decrease in pH is able to change the dynamics of important regions of the spike. Regions such as the receptor binding domain that you'll see, so this is the furin site, but the receptor binding domain will be here on the top, and also the N-terminal domain here, which is a domain that contains an antigenic supersite. And this means that our work has now provided new insights into how changes in acidity, they can modulate, they can change the dynamics of the protein, and this way may impact infectivity. Now, more recently, we've been using the DNMD approach to understand how glycans are sugars that cover, and they cover the exterior, the whole exterior of the spike. So we know that the glycans, they have important roles. They have roles in protection of the spike, but they also have functional and structural roles. And in this case, we use the DNMD approach to understand how the presence of these sugars affect the internal communication networks that operate in this protein. And once again, our results were really interesting because we were able to show that although the presence of glycans, so the glycans per se, they do not change the internal networks in the protein, their presence can either increase or decrease the response of specific regions. And this means that the presence of glycans can either strengthen or weaken certain connections within the protein. All right, and I'm now going to show you my final example. I promise this is the last one. And my last example focuses on beta-lactamases. So beta-lactamases are bacterial enzymes that are capable of hydrolyzing beta-lactam antibiotics, a specific type of antibiotics. And because they hydrolyze these antibiotics, they make them inactive. So they stop functioning as they should. So beta-lactamases are the primary mechanism for resistance to beta-lactam antibiotics in gram-negative bacteria. So we also know, well, experimentally know, that beta-lactamases, such as this one here shown here, beta-lactamases, besides the catalytic site, which is here shown in yellow, where the hydrolysis of the antibiotic occurs, the catalytic site, so beta-lactamases also possess allosteric sites, such as this one shown here in red. And allosteric sites are sites that, despite being different from the catalytic site and very often very distant from the catalytic site, somehow they're able to control, to modulate what's happening inside the catalytic site. And in this case, we use the DNMD approach to identify, to map the internal communication networks that operate into clinically relevant class A beta-lactamases, namely TEM1 and KPC2. And indeed, for both cases, we were able to map these networks and identify all the residues involved in this. However, and then we decided to take a step further in our work, and we decided to map all the clinically relevant mutations for both TEM1 and KPC2 in the structure of the protein so that we could see where the mutations lie relative to the pathways that we've just identified. And this is where things became really exciting, because we were able to see that 50% of all clinically relevant mutations for TEM1 and 60% for KPC2, the mutations lie directly onto the allosteric pathways. And this is exciting because this is a good indication that the pathways that we're capturing using the DNMD approach, they are of biological relevance. And just to finalize, it's really, really quick. So very recently, we've now been using the DNMD networks, the information that the DNMD simulations provide, to be able to identify sites, positions in the structure of enzymes that are positions that are really far from the active site, but that can modulate, control the activity of the enzymes. And in the case of KPC2, our simulations highlighted this position here, position 89. This is very far away from the active site, so the active site is on the opposite side of the protein over there. So, and this position, although being very far away from the active site, when mutated, is able to change the spectrum of activity of this enzyme without affecting its structure or its stability. And all of this, all our predictions were tested experimentally, so were validated experimentally. And just to conclude, so I hope I was able to show you that the DNMD approach is an effective way to study how structural changes propagate in proteins, but we can also look at other effects, such as effects of pH changes. And I hope I was also able to show you that this approach can be a valuable tool to study very complex problems such as allostery. We can map the allosteric networks, and explore how mutations impact these networks. And just to finalize, acknowledgements. So I need to acknowledge everyone involved in this work. So here's the list of everyone. The funders are here, and thank you all for listening.
We can have 1 or 2 questions. Questions for Sofia? Any questions? I got one question.
Right.
I don't have background on MD, but compared to the equilibrium MD, your dynamic MD, is how much more computational cost it will use to achieve a steady state or non-equilibrium?
So yes, that's a really good question. So definitely is more computationally demanding than simple equilibrium simulations, because we do need to run hundreds and very often thousands of non-equilibrium simulations to reach convergence of the responses, because we're interested in statistically significant responses. We're not, so it's definitely more computational demanding. However, it can provide a dynamic response, can provide insight into the dynamic response of the system, which is very difficult to obtain otherwise.
Okay, that's good. Thank you. Thanks.
Oh.